Okay. All right. Oh, hello. I thought I would relieve you from this, but welcome. Okay. So I have a question. I want you to think of your favorite protein and tell me why it's your favorite. You can have a scientific answer, you can have an imaginary answer, you can have a social answer, any answer. Those of you that were here first,
Eggplant.
yeah, why?
For me, it's the easiest thing to cook and eat,
Who can eat? Yeah,
and it's like really good sauce, pretty has one. Everything is there.
all right, cool. Well, they had a chance to think. Yeah,
I think I would go with a very versatile.
very versatile in what?
So like with this dessert with
Dessert.
like in baked goods or it goes with any kind of fried breakfast item. So it's very versatile.
Okay, cool. Well, Jason. Yeah, what Jason specifically? Well, I like to. And I don't want to do so but I do like Casey's is like more. Yeah, I think it's like more like applicable to like the food side. Okay, very good.
Doesn't bother much.
It's doesn't what?
Doesn't bother much also. It's stable.
Yeah. Well, single. It's not.
It is one hundred.
One of those that are really
Albumin.
egg.
Oh, egg white. Egg white? Yeah.
Exactly.
Only got one.
Okay, one is also.
Okay. What about you? Which protein do you like? Egg? Oh, gosh, the egg that we probably not gonna get to is maybe I should remove other protein and keep the egg protein. Oh, so versatile. Good. Okay. Cool. How about you, Miguel?
I was gonna say no. I don't know if specific no specific case in the no, but I guess, yeah, appreciate the thing, really. I don't know, I love dairy. It's,
Irreplaceable.
yeah, it's just hard to, I feel like tasting is just like really hard to replace, like you said, like it precipitates that, but not gelatin with huge, which tastes good to me.
Yeah, so casing can make it over and can make the. Yeah, very good. Yeah.
Gluten, which are like big breads.
I was hoping nobody felt gluten proteins. But,
I love
okay,
making bread and
okay. Yeah, that's why I like them. No, just kidding.
I'd say no, protein because. So, I don't know. No, I don't.
Great. I think I feel the passion. The energy, the passion for no. However.
Smells very good. Like, why?
first.
That's maybe a hot take, but I think that like when I'm using liquid for the plastic.
You only get one protein. What's that? No, I said. Well, okay, but no, one. Okay. Mateus.
I would say rice protein, just because we had a good moment working together during my academic tour.
Journey,
Yeah.
working together with.
Yes.
You work
Rice-
together
yes,
with
we
rice
were-
protein.
Yes.
You were buddy.
Yes, yeah, during like the 6 years.
Okay, all right, go rice.
Sir?
No.
Remove. And it's actually rice protein so bad when it's related to functionality.
But he likes it.
Yeah, because I learned a lot.
This is Mateo's.
Yeah. Because I learned a lot.
Okay,
this time.
good answer. All right. Okay, sorry, what's the question? Which protein you like and why? You get to choose one protein. Yes. Yeah. You love meat. You love the texture. Everything about meat. Beef specifically. All right. Very good. Steve. No.
I think, chicken, or I guess meat as well, but in chicken specifically, just because I think it tastes good in any form, like cooked in any form, I guess.
Okay. Oh, I need to send you this, thing about it. The kid that was talking about all sorts of chicken food and it's so funny. Oh, yeah. Just reminded me of that.
Yeah, so
Yeah. Don't say food.
No, it's actually whey protein, I think, because it's so easy to apply and everything. It's just salt and protein. For different
Your
protein.
dream,
Yeah,
for
my
your
dream.
protein.
So it's
Okay, yes.
She worked a lot with the way protein.
Don't you like it?
I like it, but yeah, I don't eat it that much the way I eat egg.
No, yeah, I know, but it, the reasons doesn't have to be food related, because it's much easier to work with than tea protein, right? Sarah, what do you like?
I just like cheese. So
Tea, yeah, okay, Casey, protein, because you like, yeah, because like you don't even need to cook, right? It's easy, fast, you need
What
more,
was fast?
yeah. Yeah.
I think gelatin, because I find like gel's very interesting,
Mm.
but also I think that was the first one that I like found out about that was like, oh, that's protein, and it kind of seemed like
Mhm.
magic to me, I guess, thinking of like Jello.
Yes, magic, I love magic. What about you?
I'm interested in family. Because I eat a lot of like your substitutes and dairy substitutes. And that's been in a lot of formulations recently, so I'm interested.
I might give you my friend that gives me an award.
I want that award.
Okay, I was gonna say gluten proteins, you took my thunder, because there's no other protein that has this good activity. So it's a unique creation, no other protein that you can make it behave like it. No matter what you do, or you do modification, nothing is gonna give you that bread that we're talking about. I like gluten proteins. Sarah, I forgot to ask you.
Yeah, because I chose made.
Yeah.
I think my favorite from potentially, I didn't like the dairy and dairy product is egg.
No,
So
the
those
only
are-
one, only one.
Only one, if I really want to select potential egg.
Okay.
Because I didn't like it.
God, now I have to really teach Ah, yeah, but anyway, okay, so I wanted to also say magic is for me gluten proteins are like magic. It's like God creation. They gave us wheat and nothing else has similar protein. The word magic. That's what I wanted to hear, magical protein. And that's why we're here. We're gonna study all the magic stuff about proteins. Okay, before I go into this lecture, I want to just go over the presentation. Again, like I said last time, we're gonna talk about it a little bit. And also, I wanted you guys to start selecting your topics. I already have 2 people that selected topics. Yeah, and then first come, first serve. I just want to remind you that we'll start with presentations April 2nd. On April 2nd, we'll have 2 presentations on the topic of protein fractionation, purification, and impact of that fractionation and purification on structure and function. We will have a section in this class talking about protein isolation and characterization. So that would fit. There are a lot of research done in that area. So that's why we are gonna give 2 students this topic. But you can tell me that if you want to choose this topic, and then you start looking for the papers. And I want to remind you that there are selections on the web, on Canvas, for you to select from that might fit the topic of your interest. So the sooner you select the topic and you finalize it, you can start working on it and ask me questions and all of that. Yes.
If we already have an idea, can we just email it to
Or before you leave, you can just tell me what the topic you want. So that's the first topic. The second topic and the third will be on April 9th. So one presentation each. So the topic 2, we're going to cover, there's a section on enzymes. So we will cover that topic, and we want you, if you select that topic, to select a paper that kind of, what I said, make sure to enforce concepts covered in class. So we're going to talk about enzyme kinetic, but how does this apply to food enzymes, basically, and the use of these enzymes? Doesn't have to be the protein, doesn't have to be the enzyme used for protein modification. It could be enzymes like polyphenol oxidase kinetics. It has an impact in food. So it could be that. And I put a couple of examples on polyphenol oxidase on the suggested presentation papers. Then from topics 3 to 6, if you select any of these topics, I don't want you just to tell me I want to work on whey protein. Now you want to tell me what component of whey protein you want to talk about. Protein denaturation and impact on functionality, or protein structure and impact on allergenicity or functionality, thermal stability and solubility. So what is really the topic? So you want to work on that protein, but what particularly you want to talk about? So that if you select, let's say, allergenicity and casein, or allergenicity and soy protein, then I'm not going to have another person talk about allergenicity, for example. So we want to make sure that the topics are diverse. So we learn something from every presentation. So when we come to protein modification, it doesn't have to be any protein in particular, but you might select enzymatic modification, or Maillard-induced glycation, or co-plasma, or other non-thermal modification, biotechnology, any type of modification, and it can be any protein. It doesn't have to be a particular protein. And for that topic, we'll have 2 students present. So as you can see, all presentations are in April, so the second half of the semester. So if you choose early, you'll have time to talk to me about your paper selection. Make sure you have few options. I want to remind you, recent papers, you might find really good paper in 2016, that's okay. It's still good paper, it's fine. But the more recent, the better, and also try to select the paper published in good journal. The way that you know if it's a good journal or not, you look it up and look at the impact factor and the rank in the case science, okay? It's most probable that the paper will be good quality if it's published in good quality journal. So you don't want to waste your time on not so great paper. You want to make sure that it's a learning experience to you and to class. So when you present, you want to say, as we covered in class, this is going to enforce certain knowledge and so on. Okay. Let's see. So yeah, you can let me know if you have an idea already. Let me know before you leave, and then start looking at papers. send me a note to meet with me for a little bit to go over your selections. And once you select and start preparing your presentations, if you have questions, you can meet with me. But for sure, I want to go over your slide once with you before you present them, to give you feedback and prepare you for the presentation. Okay. So duplication of topics will not be allowed, not because I'm just being really difficult. It's just we want to learn different things from your own perspective. So we want to make sure we have diversity in the topics we select. And I think I went over that. The first topic, we can have 2 presentations. The last topic, we can have 2 presentations, but for all of the topics in between, we do 1 present. All right. Questions? We're good? Do you have questions about that? All right. And then in the syllabus, remember, there's more detail on that presentation requirement as well. Okay. I don't know where my mouse is. Okay, there it is. Any questions, any topics from last time you want to bring up before we go to continue? So every time I start with some recap questions. We didn't go very far, so that's why I grade out most of them. Let's go from the top. What are some functions? It requires an S. Some functions of proteins in different food systems. We talked about several last time. Do you have? Yeah. Okay. Foaming. Yeah. Emulsification. Thickeners. Yeah. Through viscosity. Yeah. Dilation. Well, you don't get points for participating. You're letting people. Yeah. What else?
Water holding.
Water holding related to forming a 3D network and holding water. We're going to talk about water binding and water holding. They're 2 different functionalities. What's that? Solubility. Who doesn't get? Solubility. What else? Texturization. Absolutely. To make them texture-wise like meat, right? But they still like beef better. Okay. Yeah, there are a lot of functions for protein. These are all physical, chemical, or techno-functional properties, but also proteins can be nutritious or have nutritional benefits. They can have physiological benefits as well, especially hydrolysate, for example. They could be bioactive peptides, they give inflammation. Oh, encapsulation is another functionality. It's forming the film that will help with encapsulating flavors or antioxidants. Why are plant proteins challenging to work with? Yeah, solubility is one thing, and if they're not soluble, pretty much all the other functionalities are going to be really not great, except gluten doesn't have to be soluble, it's viscoelastic, but other functionalities. So it doesn't have, for viscoelasticity, the protein doesn't have to be soluble. For texturization, the protein doesn't have to be soluble, very soluble in water. Okay, what else?
Large molecule.
Huh?
Very high molecular weight.
Very high molecular weight makes them
Not easy
difficult to
to
work
get.
with.
They form aggregates when they are processed.
You say large blobs, large aggregates, and it's when you have large aggregates, they're definitely not soluble. They're not going to form this nice 3D network in gel formation. They're not going to emulsify very well because they're large. Movement to the interface would be difficult. What does that interface mean to you when you hear the word interface in an emulsion? You can answer.
No, I didn't hear that.
Okay, so what does, when I say interface, what does that mean to you, an interface in an emulsion system?
Oil and water.
Yes, it is the tension between oil and water. So there is this interface that the protein tries to stabilize. So otherwise you'll get phase separate. So when you have oil and water and you have a protein that is a blob, it's not going to be easily moved and form a film around the oil, or if it's water and fat around the water to stabilize it. Okay. So there are potentially a lot of challenges with plant protein, but we love them no matter what. Have faith and hope in them. Okay. So we talked a little bit about role of protein in terms of muscle building and muscle regeneration, and we also talked about muscle degradation with aging. So it limits consumption of protein, helps with keeping our muscles as we age. So that's one thing. But we also talked about nutritional contribution with proteins and having essential amino acid, and also it has to be digestible so we can benefit from it. And just we talked about that last time, and we talked about PD class, which is a measure, a combined measure of how digestible the protein is and how complete the protein is in amino acids. So most of the animal-based proteins have high quality score, and soy protein is pretty much comparable. However, it goes downhill from there with other plant protein sources, and it's mostly because of limitation in amino acids. But also when those plant proteins are processed, the digestibility score goes down as well because of them being large proteins and they form large aggregates, then our digestive enzymes will have a hard time digesting them. So digestibility is impacted by processing as well. All right, so we talked about this last time. We didn't talk about physiological functions, but briefly muscle growth regeneration, prevention of muscle loss with aging, and then in general, protein is associated with good health in general, unlike carbohydrates and lipids, for example. So protein so far, but of course, everything in excess will cause a problem. What happens if you only eat protein and a lot of it? Kidney function. No. Everything in excess is not good for us. And we talked briefly about bioactive peptides. So sometimes we generate bioactive peptides through natural digestion, and these peptides, if they are 2 to 3 amino acids, they can be absorbed directly and they will have physiological functions. Some of these functions include reduce hypertension, antidepressant, promote satiety, antioxidants, that means anti-inflammatory. And sometimes we produce them outside of the body. Some of the hydrolysates that are produced, protein hydrolysis, they claim that they have some bioactivity as well. So soy protein in specific has an association with another physiological benefit that it is associated with after FDA did so many studies on hypertension. It was linked, the consumption of 25 grams of soy protein was linked with reduction in heart disease or risk of heart disease. So this came out in 1999, and when FDA gave a health claim out, this is a result of many studies, long-term studies to confirm that. And I don't know if any of you heard 2 years ago, FDA wanted to remove that. We heard that. Did you hear about that, anybody? So that caused a problem with food processors and ingredient producers. It's like you can just delete a claim without having studies to show the content. So the claim persisted, so it's still there. Now this association with isoflavones is either considered good or bad. The literature gives you contradictory results. Some say it's not good to simulate estrogen, so it is not good. Others says, well, as if they don't have physiological benefits like antioxidant, anti-inflammatory, so good for us. So I don't have an answer. It's just considered a good source of isoflavones. What without platinum. All right, but the most what we can focus on is the physical chemical characteristics of the proteins. So color, we're not going to focus a lot on it or at all, really, but protein plays an important role in food coloring, especially casein, the cheese. So we have this white color because the caseins reflect all colors, so give you light, white color, the light scattering. And then myoglobin is a protein, red in color. Betalin in red beets is a protein, red in color. But we also have the browning where enzymes are involved, like a non-enzymatic reaction, which is Maillard reaction. Okay, so the Maillard reaction, which is basically reducing sugar and a primary amine, then it gives you this nice characteristic brown color in the bread, but sometimes too much of it is browning, like in high protein beverages. If you put reducing sugar, like high fructose sugar over time, you're gonna get brown color, which is not desirable. Now, these two examples here is due to what? The browning in an apple and the browning of tea. Polyphenol oxidase. So the browning in an apple is not desirable. And then, do you know Granny Smith apple? You know what they did to it? Genetically modified it to have very low concentration of polyphenol oxidase. So when you cut a Granny Smith apple, it stays white or light in color longer time than any other apple. Tea, you want this browning in tea. It is caused by polyphenol oxidase, but it is desirable in this case. All right, so flavor, hydrophobic amino acids are bitter. So that's why when we hydrolyze protein, a big problem with protein hydrolysis is bitter, because a lot of the peptides, most of them, if they have high percentage of amino acids that are hydrophobic, and then you release them, then we taste them as hydrophobic, bitter, or we taste them as bitter. So that's not desirable, obviously. And also when we hydrolyze, sometimes we have sourness, especially if we free a lot of amino acids here. Then we end up having some sour taste. Cysteine, like your favorite protein eggs, is high in cysteine, so it gives you sulfur taste. Monosodium glutamate gives you the umami flavor, which is the glutamate from glutamine or glutamic acid interacting with sodium gives you the monosodium glutamate, which has the umami flavor, which is desirable in some applications. Then proteolysis during cheese ripening can also give characteristic of flavor. So cheese ripening causes proteolysis and lipolysis, so lipid hydrolysis and protein hydrolysis. Both can give you those characteristic sharp flavor in ripened cheese. And then sweetness, an example, of course, that you're all familiar with is the aspartame, which is an aspartic acid peptide with phenylalanine. So it's a peptide. So it's 2 amino acids, aspartic acid and phenylalanine gives you this peptide that is very sweet. So, well, that's about it for this. We're going to spend a lot of time talking about the physical characteristics of functionality, basically. So the functionality or physical functionality of the protein impacts the texture in foods. So water binding impacts viscosity. So if the protein has high water binding capacity, it will form viscous solution. So water binding impacts viscosity. Gelation, coagulation, there's so many different gels in food system gels. So you have the jelly with your magic protein gelatin, and then you have the tofu is a gel, then we have yogurt is a form of gel. So, and then of course the hot dog is a gel. So you have different foods that have the gelation capability by forming a 3D a network of protein that can hold water. So water binding is different than water holding. So water holding, you're physically holding water in a matrix. So you form the matrix and the water is entrapped in. Whereas water binding, each protein molecule is actually binding some molecule of water. So that is the difference. Unfortunately, in literature, they use them interchangeably, which can cause confusion. A lot of time they call it water binding capacity when they actually mean water holding capacity. But you're already learning and you'll learn more about it. So you will be, when you're reading a manuscript, if you read water binding capacity, you can say, oh no, they mean water holding. So the way that you're going to differentiate them is literally holding. Your gel is holding water. So you need to have a 3-dimensional network that will hold on physically in trap water. They use them interchangeably afterwards, like wrong. So sometimes they mean water holding, but they call it water binding. Yeah. Oftentimes that water binding capacity is used wrong. Okay, and then of course, solubility is important. It impacts, it's a textural property because if the protein is not soluble, when you shake your protein drink, and especially when you have glucose beverage, actually some protein beverage on the bottle, they say, shake it well. No, really shake it well. Have you ever read that? So shake it well. No, really shake it well. So you have to shake it so that you disperse that protein, and then you drink it. Then you can sometimes feel like a coating texture on your tongue, like rough coating texture, like you have chalk in your drink. So that's why solubility is actually impacting the texture of it. So the beverage, however, when you drink whey protein, that's teaching often. They say that they feel it like whey. When you drink a whey protein acidic beverage, you don't have that coating or that chunky chalk, but you have another sensation. What is it? You probably had the whey protein.
I was gonna say sometimes you do that chocolate.
Chalkiness, is it? Or is it something called astringent?
Oh.
Chalkiness. Yeah. So you have like, I don't know how to describe astringent. You have like something catches in your tongue. Like it is, yeah, I don't know. But it's not coating like a chalk. That's, what do you think?
It is not a good blend if you're drinking.
Oh my gosh.
We're not talking about dance. And we are recording. Okay, so So when you drink whey protein beverage that is acidic, you have that acidity and sourness things. But if it's a neutral beverage, whey protein have very nice feel to it. Whereas pea protein or any other plant proteins, it's very hard not to get that chalkiness in it. It just gives you that weird texture. And also over time, you get either sedimentation or gelation over time. So really, it's a very, very challenging to produce high protein beverages. Okay, emulsification and foaming. So what is ice cream?
Foam and
Okay, foaming. Yeah, it's both because you're not fat-free ice cream. Your regular tasty ice cream that we all love has a lot of fat in it, a lot of sugar. Well, that's a good ice cream. If you want to get sugar-free, fat-free ice cream, don't eat ice cream. Okay, so yes, so what you're having here is you have your aqueous and your fats. Of course, you have your emulsion happening with the, if it's dairy, of course, you have your dairy protein helping out, but also you form a foam. So you're also holding foam. So ice cream is an emulsion and foam. What about sponge cake? Okay. Foam and emulsion. Because of course, not fat-free cake, the regular cake that has all the fat and everything that makes it taste good, you're actually also having an emulsion, and then when you bake it, you're creating that foam as well. After you beat, yeah, I mean, the cake, don't you beat it? Right? You do beating. I'm not a baker, I'm terrible baker, so I know that you beat something. Okay. And then of course you have the magic protein, the viscoelastic properties that's only present as it should be in wheat. Okay, so what caused all these structural differences? Really, so protein doesn't exist in vacuum, obviously. So you have interactions, chemical interaction happening. So of course you have protein-water interaction if the system is mostly water. So you have protein-water interaction, you also have protein-protein interaction. So that's bad in a beverage to have a lot of protein-protein interaction that will cause sedimentation, high viscosity, degradation. You don't want that, but it could happen. So you have protein-water interaction, you have protein-protein interaction, you have protein-lipid interaction, protein-sugar interaction, and so on. So you have all of these interactions that will give you a functionality. So it gives you a dilation, it gives you an emulsion, it gives you a foam, it gives you a soluble, a solution. And all of these interactions, they're chemical interactions. You have ionic interactions, hydrogen bonding, hydrophobic interactions, van der Waals as well. And then you have covalent interactions. These covalent interaction causes cross-linking. So you have protein-protein interactions through what covalent bond? Did anybody remember or know? When a protein forms a covalent bond with another protein. What is, Yes, disulfide bond. That's not only the covalent bond that can happen between proteins. We're going to give other examples of other covalent bonds that could happen, but most common in protein is a disulfide interaction. But covalent cross-linking can also happen. May not be. It's not disulfide. You have a covalent bond. It's a condensation reaction between a sugar and a protein. Okay. So we're going to study all of these interactions bit by bit with a lot of examples. Okay, life is not perfect. So proteins have their downsides. Skipping. No, I'm just kidding. No. Well, So enzyme inhibitors are proteins. So trypsin inhibitor is a big issue in soy. That's why you don't eat a lot of raw soy, because that gives you digestion issues with the protein because it will inhibit trypsin in our gut. There are also amylase inhibitors, and these are also proteins. So enzymes are protein, most inhibitors are protein. Okay. Allergens. So we used to have big 8, now it's big 9. So what did we add? That's it. So these are your big 8 and 9. Mostly they are because of the protein. And they are big 9 because you know why they are labeled big 9? Or they were big 8, not big 9. Why are they the big? Are they the only allergens? No. Huh? Consumption? Well, I'll get to consumption. You have a point here, but I want to say why they are labeled big. Death? Well, not really. Well, there are severe allergies. Doesn't have some people have severe allergies to strawberries. Did you know that? Huh? Not well. I'll come back to consumption. So over 95% of allergies in the population is coming from it. So most come, yes, that's why they are big nine. Now, get you to the consumption situation. What's going to be big 10? Yes. Sorry, I'm sorry. Maybe it's starting to change your topic. So I don't know. It's just consumption, exposure in a society that is not accustomed. So 50, 60 years ago, there was no soy protein out, especially in the West. But all of a sudden, soy protein was in everything. So people got exposed, sensitized, started developing allergies. So it is a matter of sensitization, exposure, sensitization, and then development of allergies. There are people allergic to food already, but they didn't make it to the top 10 yet. But yeah, I had a student that was allergic to peas flour. So, so that's it. But some of these allergies, some people, they grow out of it. So soy is especially in kids and they grow out of it. But peanut allergy, for example, one that hard to grow out of. Okay, well, that's not wonderful. See, the egg is there. Okay. So that's it. All I have bad things to say about protein is done behind us, moving forward. All positive stuff from here on. Yeah, you can. Of course.
People can look out allergies when they're already. What makes it feel?
Yeah, you want to talk to an immunologist. I really don't know why they no longer recognize it as an antigen. There is a desensitization. So there is sensitization and desensitization. I worked with a doctor, immunologist at Mayo, for a project that we had. So I was hopeful that I can eliminate the allergenicity of pig soy protein at the time. So the results showed that some patients we got serum. Some serum, the immune activity reduced when we modified the protein. However, immune activity was increased in other serum. He said that although this finding is not like what we wanted, but we can use this to desensitize allergens. So maybe over time, when they get a little exposure of it, by random, by not knowing, they get desensitized. But who
The
knows?
basic idea is just get exposed.
Yeah, but some people you cannot desensitize. They have severe allergy, they'll die. You cannot desensitize. That's why I'm in food science, not medical. No. don't have a great answer for you, but yeah. Okay. So we're going to study protein structure and we're going to limit it to functionality. And we are going to translate that to different proteins. So we're going to cover a lot of structural characteristics and how the structure characteristic is native. That means this is how the protein is made. And these characteristics, like the protein source with a different amino acid composition and sequence, has some ionizable groups, some reactive group, like some fiber groups, that will impact their molecular interaction, their hydrophobicity on the surface, their total hydrophobicity, their conformation. What is complement? Arrangement. So you have tertiary structures, you have quaternary structure, you have random like globular structure, you have open structure like casing. Who likes casing? The characteristic keys is due to their very unique open structure. Anyway, so you have those native, we call them also intrinsic factors. They're very unique to different sources of protein. So, newsflash, Andressa, pea protein can never be whey protein. You can never make a pea protein behave like whey protein because physiologically they're different. You cannot. Whey protein, why is whey protein produced in the milk? In the cow's milk or sheep's milk? Why do we have whey protein? It has specific function for the growth of the cow. It's not for us. The cow doesn't care about us. Newsflash. And they're like, what? Disappointed in all the cows. But so it has a nutritive function for the cow. Whereas the pea protein is a storage protein, has a specific function for the growth of the pea. Physiological. So that's why we have intrinsic factors that makes the protein unique. That's why we're going to look at different proteins and different sources and how they are different. But we don't tend to eat the protein Mother Nature intended. So we process, first, if they are plant protein, we try to get them out of the matrix and we do a lot of changes. pH, temperature, salt, ionic strength, and then we do heating through pasteurization, drying, extrusion. We use extraction solvents like hexane to get the fat out. We use other acids, bases. So we do a lot during our process. We also store different humidity, different temperature. Then we also modify to make those plant protein behave. We try to modify as well. So what happens is we go from a native state to the nature state. So the protein doesn't stay as nature intended. Although the intrinsic characteristic, it will impact how the protein works under different conditions. The same protein under the same condition will behave differently because of the intrinsic factor. So everything is related. Okay, so then all of that will affect functionality. So the intrinsic factor and how they change with processing will impact how the protein will function, whether it is taste or color or aroma is really when the amino acid forms volatile compounds. Okay, so and then hydration, that means interaction with water. This could be solubility, water binding, water holding. These are all related to interactions with water, whether physical interactions or chemical interactions. All right, surface properties. So emulsification, foaming, that means, the protein is interacting at the surface of the interface. And then because of the surface of the protein, it's interacting with different medium because of what's on the surface of that protein. That's why we call it surface properties. because what's on the surface of the protein is causing its interaction and the formation of the functionality. Is it charged? Is it hydrophobic? Do we have free sulfide group on the surface? What's on that surface of the protein that causing interactions with the environment? All right, and then structural properties. That's when we feel it. Viscosity, we're gonna feel it in our mouth. Whether the soup is viscous or the drink is viscous. Gelation, that's a structure. Viscoelasticity, that's a structure. Subsidization, obviously, is a structural property. Any questions? Did we decide to take 5 minutes break or keep going? Okay. I mean, you can always change what you, once we start diving into heavy stuff, you might say, I want 5 minutes break. All right, so this is the definition of a functional property. You don't have to memorize it. It's just for you to, understand when we say functional property, what are we looking at? We're looking at a physical-chemical property that affects the processing and behavior of food and how we feel it, the sensation or the texture that we end up, that would determine the quality of that final product. It's a kind of very fancy definition, really. Okay. Read and tell me. I think we all know this. Yeah, we kept talking about whey protein. Sorry if Andres says not pea protein. It is really whey protein because at this, do you know why it is acidic beverages are usually preferred from a manufacturing perspective than neutral? Huh?
You don't need extensive cooking.
Exactly. You don't need to. You pasteurize at low temperature. So then neutral foods, because of the microbes, they are not stable at this stage. So you can kill the pathogen at less temperature than at neutral pH, you need higher temperature to get rid of your pathogen. So with lower temperature time combination treatment, you preserve the flavor and characteristic of your beverage. So it's always desired to have acidic beverages than neutral beverages, but whey protein, as of yet, is the only protein that I know of that will form a clear, transparent beverage at pH or 2.5 to actually 3.5, which means clear. Other proteins, it's very close to their isoelectric point, so they become cloudy. So they don't give you that nice, clear beverage. And it's a very soluble protein. So whey protein is the golden standard for acidic high protein beverages. Okay. A muffin ingredient declaration had sodium caseinate. Why was sodium caseinate chosen? It's not naturally in a muffin recipe, but it was added. What do you say? It's very good. It's an emulsifier, but it also have high water binding. So it also causes the batter to be more viscous and rich. So increase viscosity of your batter, enhance the emulsification properties, and that will lead to nice texture, smooth texture of your baked muffin. So we're going to start. So this is kind of the map that this original slide with all of these boxes is the map of the class. So what we're going to start with is talk about structural characteristics as impacted by intrinsic factors. Then we're going to talk about how these structural characteristics are impacted by processing and the environment and how that impacts functionality. Then we go to individual proteins from different sources and study those. So that's kind of like the layout of the course. So we're going to start from the very beginning, and I'm sure all of you at some point in life studied proteins, right? Which courses? Let's see. Which courses you were introduced to proteins? Yeah, which comes? Organic chemistry. When you studied about the stuff like biochemistry, food chemistry, intro to food science, ingredient functionality.
Oh, yeah.
Yeah. Okay, so I should pack and leave at this point, since you have seen proteins from different classes. But I'm going to start with you from the very beginning, because this is how we're going to build our knowledge. So we're going to talk about amino acids again. You're not going to like this. You're going to learn structure of every amino acid. You like this, right? I felt the excitement. Well, they're not very hard because they all follow similar. They are a little different in the R group. And once you know the R group, you're going to understand every interaction later on we're going to talk about. Very important. Okay. So proteins differ in amino acid composition, and not amino acid composition, only the sequence matters. So even if two proteins, X and Y, have the same amino acid composition, you know how you determine amino acid composition, or you probably heard of it one time, is you break all the bonds. So you have 3 amino acids and you quantify the content of each amino acid. Then you get the amino acid composition, but this doesn't tell you anything about the sequence, because you break down all of the peptide bonds and then just determine the quantity of the peptide. So that's what I mean by amino acid composition. So some proteins are different than amino acid composition, but not only that, the sequence. So what amino acid comes next in the peptide, in the whole peptide? So that is very essential, and proteins differ in sequence. And because they differ in sequence and amino acid composition, they are different in configuration, how they fold, and what form they take, and whether they are only a tertiary structure or they form quaternary structure and multiple polypeptides again. So the composition and the sequence determine the structure of the protein, the configuration. Also, the amino acid composition and the sequence determine molecular interaction. That's why we need to learn amino acid R chain, because the R chain have different functional groups that will either interact more with water, or interact more with fat, or do protein-protein interactions, or hydrophobic interactions. So the amino acid composition and sequence will determine these molecular interactions, whether internal within the protein or outside of the protein. And then the chemical biochemical properties. So a lot of the folks in my lab will always measure hydrophobicity and surface charge, surface hydrophobicity and surface charge. Why do we care? Because these are chemical biochemical properties that will determine how this protein is going to interact with others in other molecules in the environment, and how would that impact function. So that is a very important property or set of properties, and they are also dependent on amino acid composition and sequence. So this amino acid composition and sequence dictate a lot of things.
Determine the amino
That's coming in lecture 3. No, they determine amino acid sequencers. Either you can do that with mass spectrometry. We call it ladder mass spectrometry, and I'm going to talk about it, or something we call adenine sequencing. Again, I'm going to talk about that in lecture maybe 4. Yeah. So, so since different proteins from different sources are different in these characteristics, they are different in their functional contribution. And the environment impact these characteristics. The environment might not impact the sequence per se or the composition, but it might change charge or cause molecular interaction, polymerization, for example, or more like water, a protein fat interaction, or protein carbohydrate interaction, and so on. So, intrinsic factor such as amino acid composition and sequence cannot be impacted by processing and changes in the environment. You might break the protein into small molecular peptides. Yeah, you are breaking the sequence in that case. But if you're not hydrolyzing and breaking it off, really nothing you do during processing can change the amino acid composition and the sequence. Unless you degrade your amino acid, like calcium is degradable under harsh conditions and presence of sugar. So you can degrade some amino acid, but you don't really change the composition and sequence. So bear that in mind, but you can change everything else with processing. The configuration, molecular interaction, the surface properties due to the environment, and thus you can change something. Basic. You've seen this, I'm sure, before. First of all, what is pKa? What does it mean? Probably heard it in chemistry, general chemistry, food chemistry, food analysis. Yet we hate this term so much that we forget everything about it.
It's some sort of equilibrium.
Sort of equilibrium between what? Yes, say one word, and I will say if the word is present in the answer. Huh? Acid? Acid base. Huh? It is related to the charge of the protein. You're all saying correct things. Now put them together. Okay. It's 4 p.m. Wednesday. It's the contributing factor. Okay, so the pKa is the pH at which your amount of dissociated groups equal to the concentration of undissociated. So so let's say we're just talking about carboxyl group. What is carboxyl group? COOH. At the pKa of that H, the dissociation usually, the dissociation factor is the H in this case. So at the pH equal to pKa, half the concentration is CO minus and the other is COOH. So that's the equilibrium. Somebody said equilibrium. You have same concentration of carboxyl non-dissociated and carboxyl dissociated. So COO minus concentration equals COOH. Now, but the protein is more complex. You don't only have carboxyl groups, you also have amine groups. So you have pKa1 for acid groups, and you have pKa2 for amine groups, basically. So the amine group is your NH2 or NH3 plus, right? NH2 or NH3 plus. So pKa2 is the pH at which you have same concentration of NH2 and NH3 plus. There are drawings that we will see later on. Okay, knowing that, let's see if you can guess the answer. Side chain, okay? So in the side chain, some amino acid have a acidic side chain, some amino acid have basic side chain. Do you know if lysine, arginine, and histidine are acidic or basic? Basic. Okay, it's fine. You don't remember. We're going to remember all of this together. So they're basic. So your pKa is lower or higher than high. So none of them. Side chain. Side chain in basic amino acid has amine group. Okay, so it's basic. So your pKa will be basic. In order for the amine group to lose its proton, the pH needs to be high to give you its proton. Okay? So above, so the pKa will be in the basic range. Okay. All right. Let's just a check, knowledge, refresher. All right, so this is your amino acid. You have your chiral carbon. Chiral carbon mean it, so the carbon makes 4 bonds. So chiral carbon means each bond has a different group attached to it. That's all what it means, chiral carbon. So it has a carboxyl group, COH. It has an amine group. It has an H, and it has an R. R is your side chain. So every amino acid has carboxyl group, amine group, side chain. Easy so far, right? Let's add on. So basic, acidic, buffer. You've heard that proteins are good buffers because as you change the pH, either it will pick up a proton or give you a proton. So it buffers the pH. So if you're titrating with an acid, your carboxyl groups aren't going to pick up protons. If you're titrating with a base, your carboxyl groups is going to start giving, well, of course, they will be the first to give you their proton because they're acid. And then the amine group will start giving you their protons as you increase the alkalinity. Okay, so buffer. They buffer. You have the 2 different charges there. They buffer the pH. That's the Zwitter ion definition comes in. Zwitter ion, where you have a positive and negative. So you have a dipolar molecule with acidic group and a basic group. Okay. So you have a positive charge and a negative charge. So that's just reminding you of what is Zwitter ion. Okay. The R group is the most important group because it dictates interactions and therefore it dictates functionality. That's why we're going to have to study all the amino acids because in order to understand interactions, we want to know what's interacting with what. So the R group, it determines polarity, interaction with water, hydrophobicity, if it's hydrophobic, charge, if it is carrying an ionizable group, and thus the interactions. And of course, leading up to functionality. So this is a great slide to study from. So let's dissect this slide a little by little so that it makes it easy to swallow. So I can't see from here really. It's like breaking your neck to look up there. Okay. So if you look at the top, so the red is aliphatic. So there's also the solid red, and then there's the dotted. Well, not only solid. So there's the solid ring, and there's the dotted ring. So the dotted is for essential amino acid. So everything dotted here is essential amino acid. And then the solid red, or just red in general, it's aliphatic, meaning the R group doesn't have a ring. Doesn't have a benzene ring, doesn't have a phenol in it. Okay, so aliphatic. Then aromatic. So these, we only have 3 aromatic amino acids, the phenylalanine, tryptophan, and tyrosine. You have a ring structure here. Benzene, basically. Benzene structure, and this is a phenol. Okay. Then acidic. Where are our acidic? These orange, I think that is aspartic acid, glutamic acid. These are acidic. And then we have the basic. We have 1, 2, 3. Arginine, histidine, lysine. Then hydroxylic. That means we have a hydroxyl group. OH. Serine, threonine. They become really important later on. You will see why. Not only these, I mean all of the rest. Sulfur containing cysteine, sulfonyl group, free sulfonyl group, very important for protein-protein interaction and stabilization of the tertiary structure and quaternary structure. And then we have methionine. Amidic. So asparagine and glutamine, they have an extra amine. And well, this amine group, if you add high acidity or high alkalinity, what will happen to asparagine and glutamine? Under high acidic condition or high alkalinity, what do they become? Yeah, aspartic acid and glutamic acid, they lose their amide group here. So when they lose their amide group, this becomes a carboxyl, this becomes here a carboxyl group. So these two become aspartic acid and glutamic. So these are the amines. Okay, so the other thing that you want to look at is the 3-letter amino acid. So most of you know these 3 letters, be good refresher. I'm not going to ask you to learn the 1 letter. It's very confusing and doesn't make sense. Why glutamic acid is E? I don't know who made that up. There's no E in there either. I know. I have no idea. That's why it doesn't make sense. So alanine A, okay, makes sense. Glycine G, okay. Why aspartic acid is D and why glutamic acid is E? First come, first serve. First come, first serve. I've taken my, I think the A is a patient. No, don't confuse me. I really don't know why the 1 letter is based on what. That's why I don't ask you to know the first letter. But the 3-letter makes sense. You'll see that when you look at the 3 letters, it's easy to remember, it makes sense. Okay. So we're going to look at all of these different amino acids and see how they are different in their R chain. So hydrophobic. All aromatic amino acids are hydrophobic. They form hydrophobic interactions very easily. And another really nice characteristic that we use in analysis is that they absorb UV light. So all of them absorb UV light, but mostly phenylalanine and tyrosine have the highest extinction coefficient. It might bring back nightmare from the analysis if you're taking the analysis. But bless you, Emily, you didn't take the analysis. That was on sabbatical. It was your loss.
It was.
She can't say anything else. Yeah. Okay. So higher molar extinction, meaning they absorb more, their absorptivity is high at 280. Tryptophan does absorb, but not as much. So this is very helpful characteristic in determining hydrophobicity, for example, and using fluorescence. So this becomes really important, these components. Every protein has a certain amount of these 3 amino acids. So every protein can be analyzed. And if you have a protein solution, no matter what is that protein solution, you can measure absorption at 280, plot the standard curve and get protein concentration. Or if you are running HPLC and using UV absorption or UV fluorescence for any protein, you can do that because of these existing in all proteins. However, if you are analyzing peptides, not every peptide will potentially have any of these amino acids. When you break down the protein, you have peptide. That's why you cannot really measure peptides at 280, then you can measure at 220, which is the wavelength where peptides absorb light. Okay, so the characteristic here is their hydrophobicity and also their ability to absorb and fluoresce light. Another important characteristic for tyrosine specifically is a phenol. So this group becomes ionized at high purity, meaning it will carry a negative charge. That's why a lot of the protein, most of the proteins will have higher solubility at alkaline pH. Because now we're having not only the carboxyl group that is charged, negatively charged, but this phenol becomes negatively charged, so then the protein becomes more soluble. That's why when we cover solubility, we will see that the solubility curve for plant proteins, most plant proteins will look like that. So keep that in mind for later. It will come back later. But I don't know, I can go as low as this, I guess. So here you have the solubility, sorry. Let's say on the y-axis we have solubility. Here we have pH. And then most of plant proteins have this curve, right? So this is, let me make it a little bit more dramatic. So let's say this is pH 10, this is pH 2. So at very low pH, you have net positive charge. So you have good solubility. Here, around the isoelectric point, so it has lower solubility. And then at alkaline pH, you get that extra negative charge. Depending on the composition, amino acid composition, the more tyrosine you have in there, the more negative charge you're going to carry, the higher is the solubility at alkaline pH, mostly for plant proteins. Say that again. If protein, yeah.
The protein goes the second one.
Yes, because then if the protein, if you are centrifuging at the isoelectric point of the protein, you put your solution at the isoelectric point of the protein. The isoelectric point means your net charge of the protein is zero. So you don't have as much interaction with water. You will have more protein-protein interaction. There's no more interaction. So protein is one interact with protein, and then before it is established at the isoelectric. And then when you measure solubility, you can take the super and see how much protein you have. That's a measure of our okay. Okay. Then we have the nonpolar or hydrophobic, but instead of ring, they are aliphatic. So there is no ring structure. Proline is unique. It has a cyclic structure, but not a ring. So there's no benzene ring there. There's a cyclic structure. It's very unique. It's the only amino acid. See this amine group? And this is carboxyl group. It's the only amino acid that the R group is actually covalently linked to the amine group. The only amino acids out of 22 that the R group is covalently, so the carbon, the last carbon in the R group is covalently linked to the nitrogen. So that is a very unique amino acid. It's not a benzene ring. We call it cyclic. Not a ring, cyclic. Oh, So these are the amino acids that are aliphatic and hydrophobic. That means not liking of water. Methionine is unique because it has a sulfur group here, but the sulfur in here is not an SH, not a sulfide group. It can form two bonds. Already formed the two bonds with a carbon here and a carbon here. So there's no SH. So it's not a free sulfide group available for disulfide chemistry, already available without breaking some. Okay. Just a side note, you don't have to remember, but isoleucine, it has two chiral carbons. One, the one that is bonded to the carboxyl group and amine group, which is chiral carbon alpha, and then another carbon, chiral carbon beta, because also it has 4 different bonds. This is with a methyl, this is with the ethyl, this is with an H, this is with a carbon. So that's why, but it's not that important. I just wanted to point it out. So this is unique. It has it's called secondary amine because usually your amine is a primary amine. The amine group at the end that is not reacting with anything is primary amine. It has one covalent bond with carbon and the rest are hydrogen. This is a secondary amine. And it's very important in the formation of secondary structure. Once we talk about secondary structure, you're going to hear me say it is, I'll say it again later, a secondary structure breaker. In casein, so you like the casein protein, it's very unique in its content of proline. 17% of amino acids in casein are proline. That's why most of the structure, secondary structure, is random coil in casein. So open structure, random coil, because the proline, because you don't have a lot of flexibility of turning, it won't allow you to turn and form, your alpha helices or beta sheets or beta turn. It just, we call it secondary structure breaker.
I thought as one. We have a high diversity, why we have a high.
So alpha S1 is one of the caseins. So you have casein alpha S1, casein alpha S2, and then you have kappa casein and you have beta.
So generally increase the flexibility.
It generally decreases, but if you want to compare them to each other, there is difference in how flexible they are. But all of the caseins are open structure. But beta casein is the most hydrophobic, for example. And then the alpha S1, I believe alpha 1 is the one that has the most phosphorus and calcium phosphate in formation ability. So that's different. But in general, to all cases, you have a higher proline content than different proteins. So that's why their tertiary structure is very open and secondary structure is mostly random. Okay. Oh, the other one here I want to point out, hydroxyproline is a type of amino acid that is a post-translational modification of proline. It exists in meat protein. Okay, and then we have the polar charged. Okay, so there are the basic charge, meaning at neutral pH or mostly around neutral pH, they carry positive charge because of their extra amine group in the R chain. And then the acidic, they have additional carboxyl group, which will make them carrying negative charge, higher negative charge at around neutral pH. Okay, so because they carry charge and they have ionizable groups, so they are polar and they interact the most with water. Because of this additional charges in the R chain. Okay. So another amino acid that can exist post-translational modification is hydroxylysine, and that's also in liquid. Now, what's the importance of some of these amino acid? Lysine is very important. We said processing impact lysine degradation because it's the most reactive amino acid in the Maillard reaction. Why is it the most reactive? Because if we look at the amine group here, it is a primary amine. So it only has one covalent bond with carbon. The others are hydrogen and carries a positive charge. That's a primary amine, most reactive. Usually the other primary amine in the amino acid is usually bound in a peptide chain, right? Because your peptide chain is a carboxyl group bound to amine group. When you have ping, you only have one amine at the end, the N terminus, and then you have one carboxyl at the end, the carboxyl terminus of the whole polymer. So the R chains in the middle are the one that determine the activity with the environment. So the lysine will have that extra primary amine that would be the most reactive in Maillard browning, either causing desirable browning or excessive browning and nutritional loss. So if I go back once, so if I look at histidine and arginine, this is secondary amine, and this is tertiary amine. So this nitrogen has 3 bonds, this nitrogen has 2 bonds, this has 1. So this is primary, secondary, tertiary. So the primary amine is the most reactive in Maillard reaction. Okay. Then we have the hydrophilic polar non-charged. So these are the amino acids that are polar, that means they interact with water or like water, but they don't carry charge. So glycine is the smallest amino acid, and it's very unique. Its carbon here is not chiral because it has 2 bonds with hydrogen. So that's very unique, smallest amino acid. And then we have those glutamine and asparagine, they're polar. When we do acid hydrolysis or alkaline hydrolysis, they deamidate. Deamidate, they mean lose an amine group. Then they become glutamic acid and aspartic acid. That's why those of you that do amino acid composition analysis, when we do acid hydrolysis, when we run amino acid composition analysis using HPLC, we can never determine the exact amount of glutamine and asparagine. They are all determined as glutamic acid because we hydrolyze the protein using acid or alkaline into amino acids. And with that high alkalinity or acidity, we convert all of glutamine and asparagine into glutamic acid and aspartic. So these guys, serine and threonine, are very important in post-translation modification. You have hydroxyl groups. Why do we care? We care because it gives characteristic functionality to our casein in this case. Phosphorylated. So serine in caseins are mostly phosphorylated, alpha S1, alpha S2, and beta casein. So meaning this OH group is phosphorylated. So instead of having an H, you have a phosphate group. This what gives the casein its characteristic ability to form calcium bridges, calcium phosphate bridges, and helps form that nice curd that you like in the cheese and help that melting properties of the cheese. So this is a post-translation modification, but it happens at serine amino acid. So it's a very important characteristic of caseins. Another important characteristic is glycosylation. This is also a post-translation, meaning it happens in the cell post the formation of the protein or translation into a protein molecule. So what happens here, you have a not glucose, a carbohydrate moiety attached also via the hydroxyl group of threonine in this case. This is very important in kappa-casein. Why? Do you know the micelle, the casein micelle? What keeps the micelle suspended in milk? A colloidal suspension. Kappa-casein. Because kappa-casein dominates the surface of the micelle and it's glycosylated, so it's very polar and also carries a net negative charge. So it stabilizes the casein micelles in milk, so they are stable suspension, stable colloidal suspension. Because of this glycosylation, that's because caseins, they interact via hydrophobic interaction, you have beta-casein, very hydrophobic. So they form these clusters, they form these micelles because of the hydrophobicity and their open protein. So their hydrophobicity is mostly exposed. And they have those calcium phosphate bridges. And then if there is no kappa-casein glycosylation, what the milk is a suspension that you put the glass of milk and you see all of the casein in the bottom. But that's not what nature intended. So that's why we have this glycosylation that makes the milk perfect beverage. Yeah, Emily, that's what, tell me, this is why you like milk protein. That was your original thought. Okay. All right, so when we have this phosphorylation and glycosylation, we tend to have higher hydrophilicity. But in the case of phosphorylation of casein, we have a lot of calcium, so it causes the calcium phosphate bridges. But in case of the glycosylation, enhance the solubility and the stability of these micelles. Okay. What else do we have? We have the cysteine that is good and bad. It has good qualities and it has bad qualities. So the good qualities is it causes stabilization of your tertiary structure. So what happens? It's an oxidation reaction. You have your cysteine, or if you want to pronounce it, this is cysteine, and this is cysteine. The most confusing pronunciation ever. Cysteine and cysteine. Okay. So the cysteine here is actually two cysteines combined by the disulfide linkage. So this is an oxidation, but this bond can be reduced. So that's why those of you that run STS page, electrophoresis, you add this beta-mercaptoethanol, and that beta-mercaptoethanol is a reducing agent, so it will break disulfide linkages. And if you have any polymers or a quaternary structure stabilized by disulfide linkages, you add that beta-mercaptoethanol, it breaks it, and your protein migrate at individual subunits. Okay. So this happens intramolecularly. That means within the one molecule of protein, stabilizing the tertiary structure, or you can form intermolecular. So protein-protein interaction, which is very common example, is the gluten-forming proteins. So the gliadins and glutamins, when we knead our dough, put flour and water and start kneading, you're denaturing the protein and causing them to form disulfide hydrophobic interaction, hydrogen bonding, disulfide interaction is forming your nice gluten. Okay, so that's an example of intermolecular. Sometimes we don't want it in a beverage. You put pea protein or soy protein, you pasteurize, and then your protein falls out of solution over time due to disulfide linkage formation. Okay, so cysteine is very important. Free sulfide groups are very important. And they stabilize the tertiary structure. When we talk about secondary and tertiary and quaternary, it's all about containing free energy. when you make the peptide in the cell translation, the genetic translation into a protein, it has so much energy and starts folding, containing the energy by bond. So the disulfide contains the most energy, and that's a very strong stabilizing force in the formation of tertiary structures. That's why denaturation temperature is higher when you have a protein stabilized a lot by disulfide linkages. All right, what else do we have? Glutamine. It's a very important amino acid. It's one amino acid that has a great potential. Have you heard of gluten? Well, in this case, it's not meat, it's bread. So trying to make rice that you like, rice protein behave as wheat protein, impossible. Cannot, needs help. So if you take rice flour and add transglutaminase, so what that does is it takes the amine group of lysine and reacts it with the carboxyamide group of glutamine and releases an amine group and form a covalent bond. So now I'm inducing protein-protein interaction, forming better network, so I can use my rice flour to make bread with a good volume. So glutamine plays an important role in that. So that's glutamine that is here, the carboxyamide, that means carboxy group and amine group. It's called carboxyamide. Reacts with the amine group of lysine, another protein. And then with the transglutaminase, you remove an amine group and then form that covalent. But that glutamine is also important in situations where you want to enhance protein solubility. So how does that work? So basically, there is another enzyme, not transglutaminase, glutaminase enzyme. So what it does, it takes that glutamine and breaks off this amide group or amine group and gives you a charged carboxylic group. So you increase the net negative charge on the protein. So by just using an enzyme to take out that amine group, you introduce a negative charge. We use it in our lab for different purposes. One of it, hemp protein is the worst soluble protein on the planet. It's not soluble, period. So in order to get the protein out of the hemp flour, we use this glutaminase and it became more soluble. We got it out and then it's soluble at pH, neutral pH now, which is something. Okay. Questions?
So if glutaminase doesn't work for a protein, does that mean it has less lysine?
Less glutamine.
Yeah.
Less glutamine, yeah, because its composition changes. And also the structure of the protein and the ability of the enzyme to find the glutamine, So we need to figure out the best conditions for your alfalfa protein, which is, sorry, I'm glad you're graduating soon. You'll be done with alfalfa. Yeah, so every protein will have different composition of glutamine. So you might not get the same outcome. And also, depending on not only the composition, but the sequence. Where is that glutamine in your sequence and how accessible the enzyme is based on the protein structure. Okay. Good questions. All right, so I'm going to move into now acid-base properties. So I just highlighted why it's important to know your amino acid, because every amino acid has different R group, and every R group has a specific function. And knowing that will help you understand the interaction that the protein can have in different systems. Okay, now, what are you all gonna do when you go home? Yeah, start drawing amino acids. Okay, so acid-base structure. So properties, acid-base properties. So these properties are important because it impacts the charge of the protein under different conditions. So that's why we need to know the pKa and of the different amino acids and then the different proteins and the charge at different pH. So pKa is the negative algorithm of the dissociation constant. So pKa equal negative log Ka, which is a dissociation factor. In this case, it's the H. We're talking about the H, the proton. All right, so we have the carboxyl group have higher dissociation because it's an acid. It readily gives you its proton. Whereas the amine group, it's basic. It's not going to give you its proton unless the pH is really high and alkaline. Then it will give up its proton. So that's why in the amino acid you have pKa1 and pKa2. pKa1 is for the acid and pKa2 is for the base. That's basically what is said here. pKa1, pKa2. This is for the acid group. This is for the base group. And the acid group will be low. This is acid. And the amine group will have acidic, basic pKa. So what does it mean? Above the pKa, most of your molecules are negatively charged. Below the pKa, most of your groups are positively. Okay. Now, pKa3, it has to do with the side chain. So if you have a additional carboxyl group, it will be acidic. If you have additional amine group, it will be basic. Now, the other one is the pi, the isoelectric point of your protein. So the isoelectric point is the point where you have net negative charge. So you have equal number of positives and equal number of negative. Together, they give you a net zero. All right, so how do we determine it for amino acid? Usually, if it is an amino acid with only the amine group and the carboxyl group, then you add them, divide them by 2, you get the isoelectric point, which is the point at which it's 0. As you go up in the pH, you gain negative charge. As you go down in pH, you gain positive charge. Now, if you have acidic amino acid, you take the acidic groups pKa, the pKa1 and pKa3, the acidic group. If you have basic, you take the pKa2 for the amine group and pKa3, the amine group in the R chain, and that would be divided by 2, you get the isoelectric. But these are only for amino acid. Now, don't ask me how you calculate it for protein. It's complicated. You have to take chemical biology class and learn about that. It's very hard to determine. It's a lot of calculations, knowing the composition of the amino acid and also the sequence, because where it is impacts how it's impacted by the environment. But anyway, so this is from a table from the food chemistry book, Demaduran, 2017, the Fanema food chemistry book. Okay, so what this shows you is the pKa's for the carboxyl group in each amino acid. You see they are all acidic. And then pKa2 for the basic group, all basic. And then you have a side chain. So if you look at glutamic acid and aspartic acid, they are acidic, for example. So, but again, this is dependent where it is in the sequence of the protein and the neighboring amino acid chain. So this is not a value within a protein. This is for individual amino acids. You have to memorize all that. No, no, that would be all of you will drop the class. Okay, no, it's just FYI, it's for you to demonstrate that what is acidic pKa, basic pKa, and so on. Okay. So the protein charge is determined by the side chain, which is obvious. This is talking about this peptide bond, the second bit. So the peptide bond is really acts like it is a double bond. This is the carboxyl group, this is the amine group, because these electrons here, they can be shared with this carbon. And then this oxygen gets the two bonds that are shared in this double bond. So then you create something like that. And this one, because it's already when it forms double bond here, it loses both of electrons into that double bond. It becomes positively charged, so it does not accept a proton. That's why the net charge of the protein is impacted by the side chain. Huh?
Double bond equals to negative peptide.
So the peptide bond is not a double bond, it's this carboxyl group and amine group. But the electrons are not just sitting there. They move. And then when they move from this nitrogen to here, or when this electronegative atoms, they exchange the electron, this becomes a partial double bond, but it's not really. It's just with the electrons moving from one electronegative atom to another. You don't have to know this very well. Okay. All right. So anyway, this is just explaining that the side chain is what controls the charge of the protein. The side chain of the amino acid. Last thing, I just want to show you this, where the isoelectric point is. Let's say this is an amino acid, is Zwitter ion. Let's assume it is neutral at pH 7, so that would be its isoelectric point. And then as you go up in pH, you start getting negative charges. You reach the pKa 2, and above it, it would be mostly negatively charged. And then the same, as you go below the isoelectric point, you start getting positive charge. You pass the pKa1 where the concentration of dissociated and non-dissociated is equal. If you go down still, it will be all positively charged. That's just a demonstration. And of course, your favorite equation of the entire world is Henderson-Hasselbalch equation, which we use it a lot to produce buffers, which tells you here, it kind of defines your pKa. So A minus is what? Your conjugate base. So if we want to say, in this case, what's A minus for pKa1? It's carboxyl group, you know, your COO minus. And this here is when it is protonated. If we just look at the carboxyl group, concentration of the COO minus over the concentration of COOH. When you have equal concentration, this becomes 1. Log of 1 is 0. Okay, so pH equal pK equal the pH. In this case, that's when this concentration equals this concentration. This just to help you visualize what we said earlier is a pKa is the pH at which you have equal concentration of dissociated and non-dissociated, basically. All right, I'm gonna stop here. I'm losing 90% of you. But anyway, fascinating. All right, thank you. Start thinking of these topics. Yes, you had your hand up? No, you're just stretching? Okay.